Use the following information to take notes on cell transport. You do not have to write down these 3 questions. These are the essential questions that we will be answering throughout this unit. What is the purpose of the cell membrane? Why would we need to regulate what goes in and out of the cell? And how does the cell transport nutrients, waste, proteins, etc. into and out of the cell? To answer these questions, we need to start with the structure of the plasma membrane. The plasma membrane is also referred to as a phospholipid bilayer. This is a phospholipid. Phospholipids are made up of 2 parts, a phosphate head and a lipid tail. The phosphate head is hydrophilic. Hydro means water. Philic means loving. Think about how Philadelphia is the city of brotherly love. Or, for a pedophile, peda means child, like a pediatrician. Philic again means loving, so pedophiles love children in a really freaky way. The lipid tail is hydrophobic. Lipid is similar to oils, and so think about how oils fear water and they repel themselves from it. These phospholipids will make up a layer, but note at the top that it's a phospholipid bilayer. Bi means 2, so there are 2 layers of these molecules. One of these layers is upside down. The phosphate heads, which are hydrophilic, love water. Now there's a watery environment inside the cell with the cytoplasm. There's also a watery environment outside the cell, so they turn in those 2 directions. The lipid tails fear water, so they turn towards the inside, towards each other. The second part of the plasma membrane is proteins. Proteins can act as passageways through the plasma membrane. Some can also act as cell markers. Here's a better picture of the plasma membrane. This can be found on page 182 of your textbook. The pinkish reddish are the proteins. As you see, some of the proteins form channels or pumps, and these will help move materials across the cell membrane. There are also proteins that have carbohydrates, and those act like chemical identification cards, allowing individual cells to identify one another. We refer to these as marker proteins. And then there's the 2 layers of the phospholipids. Again, with the hydrophobic tails, lipid tails turning towards the center, and the hydrophilic heads turning towards the outside. Now the plasma membrane is selectively permeable. Permeable means able to pass through. Impermeable means it's not able to pass through. We call the plasma membrane selectively permeable because it only allows certain substances through it. We will cover 2 main types of cell transport, passive and active. Passive transport is the movement of a substance without using energy. Molecules will move from high concentration to low concentration. And the 3 types are diffusion, osmosis, and facilitative diffusion. Active transport requires energy from the cell. Particles move from low concentration to high concentration, and there's 2 types, endocytosis and exocytosis. You do not have to copy this down, but this is some helpful vocabulary that you will need to know in this unit. A solution is a mixture of solutes and solvents. The solute is a substance that is dissolved in another substance, such as salt. The solvent is a substance that does the dissolving, such as water. And a concentration is referred to the amount of solute in the solution. We're going to begin talking about types of transport with passive transport. Remember, passive transport does not require any energy from the cell. The first type is diffusion. Diffusion is the movement of particles through the plasma membrane from an area of high concentration to an area of low concentration. This means they are basically spreading out. Diffusion will occur until equilibrium is reached. Equilibrium is when the concentration of the substance is the same in the given space. To better understand the process of diffusion, watch this video.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.